Jodi is giving her thesis presentation today. Jodi is a master's student from lab. She earned her undergraduate CS in bioengineering at Walla Walla University in Washington State, where she discovered her love for plants. She enjoys reading, playing video games, and tea. After her master's, she plans to pursue a PhD in bioinformatics and computational biology. She's been a pleasure to have in our lab. Truly, she's an enthusiastic young scientist, and she's ready to present her thesis. She joined our lab from fall 2017. So she's been working so hard, and for us, it's a new direction that she's working on. So I'm really proud of her. So that I will let Jodi to present her talk.
Hello, my name is Jodi Callwood. I am a candidate for a master's degree in agricultural plant science at Callwood State University. My PI is Dr. Callwood, and my project is on the analysis of two divergent species to heat stress transcriptional profiles. So what is heat stress? Heat stress is a type of abiotic stress that is environmentally induced. Think like flooding or high saline soils. And heat stress is actually a combination of two types of intensities, high temperature and intense light. That can lead to overexcited electrons and denature protein and also encourage water loss. Prolonged exposure can result in loss of cellular cohesion and sunburnt leaves, and extreme exposure can reduce plant yield and also kill the plant. So what makes a plant heat stress tolerant? Well, there are some physiological adaptations that a plant can make in order to have some heat avoidance mechanisms in order to reduce the likelihood of induced stress, such as having a waxy coat on the leaves, and that provides some filtration for UV rays. Also, parallel leaf orientation is another adaptation that can help prevent heat stress induction. On the molecular level, when a plant is tolerant, we wanted to see the plant engage stress genes and also maintain its cell walls and membranes. We want to see the plant react to reactive oxygen species and also engage antioxidants to combat those levels, and also photosynthesize and maintain proper protein generation during this time period. So why are we studying heat stress? Well, the past 4 years have been the warmest years in recorded human history. These warm temperatures are creating superstorms like Hurricane Dorian, which ravaged the Bahamas very recently. In terms of heat, high temperatures are causing a health risk for humans that are putting a lot of people in the hospital due to dehydration and sunstroke. But one particular vulnerable species are plants who are not able to go inside or pay attention to health warnings. They really need something within their genetic makeup to combat heat. And we care about this because agriculture is a national security issue. Governments spend a lot of resources in order to make sure that their people have access to food. And this impacts all levels of society because growers or farmers rely on plant yield in order to make their income. If a grower sees a reduction in yield due to loss of plants, or if the plant has reduced yield, then they're not going to be able to have enough resources in order to plant the same levels for the next year. And that will ultimately drive up prices for consumers who will be paying more for food. So it really does impact all levels of society. And there are some plants that are more vulnerable than others due to very specific growing requirements. And blueberries are one such plant. Blueberries were domesticated in 1950, which in plant science is actually a very recent domestication. Part of the reason for this was that blueberries have very specific soil and temperature conditions. They like a sandy composition of soils that is in somewhere of a pH between 4.5 and 5.5. And the Department of Agriculture says that the northern hybrid blueberry, which is a type of blueberry, has fruit set growth and ripening between 20 and 26 degrees Celsius, which is a 6-degree threshold. At 30 degrees Celsius, photosynthesis decreases by 47%. So 4 degrees outside of that range, the plant is unable to photosynthesize well, and it can feed itself, and then that will in turn reduce its yield. We care about this because blueberries have exploded worldwide because of their high antioxidant content and also vitamin C. They're denoted as a superfood, and they're now being grown in places as far as China and India, which can see well over 100 degrees Fahrenheit, well outside the temperature range of most blueberries where they're comfortable. In 2016, the United States was the largest producer of blueberries, producing over 690 million pounds of blueberries, which grows over $700 million. So we're not dealing with small margins here. And a lot of people who aren't as familiar with plant science will say, well, plants are a part of nature, they can adapt. Why can't these plants just adapt? And why do we need to intervene? Well, because humans have been intervening with plants for a very long time. Humans have been selecting which plants live and die and how they reproduce for so long that plants have actually grown to kind of rely on the human intervention a bit. And the question is, can plants still reliably produce and adapt quickly enough to keep up with the human demand as well as their own internal demand? And I would argue the answer is no. And that's where plant breeders come in, and I hope to be one someday. So the objectives for this project, with all of that in mind, are to assess the response of two divergent blueberry species to 45 degrees Celsius heat stress, and to compare the transcript response of those divergent species, generating profiles and looking at, well, which one is more tolerant and what aspects of it might be attributed to that. So these are the materials for our project. On the top here is Vaccinium corymbosum. It is a type of highbush blueberry. It's native to like the border regions of Canada. It can also be found in New Jersey, not too far from here. And on the bottom here is Vaccinium darwii, and that's native to Florida. And these two species, these are actually the plants I used for my experiment, and we got these through a partnership at Rutgers University with our partner, Dr. Borsa, who one of the first things I did as a graduate student here was drive down to New Jersey, and he taught me how to propagate my materials. So these are actually the plants that I propagated that I'm using in this study. But at the time of our experiment, these plants were only between 6 and 8 months old. They didn't have a lot of leaf material on them. And so in order to optimize our experiment to make sure that we got the most out of what we had, we implemented a test process, which is how we got phase 1 and phase 2. So phase 1 are some tester plants that have a combination of corymbosum and darwii somewhere in their genetic background with one diversifying plant. And phase 2 are the corymbosum and darwii plants that we want to look further into. And the methods, they will be denoted with phase 1 and phase 2 depending on what they're a part of. So first we did some physical observations. We took pictures of the plants before and after heat stress, paying most attention to the leaves because that was very easily observable to see if there's any outward response to the heat stress conditions that we induce. And this is how we induce heat stress. This is called a growth chamber. We're able to control the environment in this machine here. And we chose a temperature of 45 degrees Celsius with a light intensity of 3, which means all the lights in the growth chamber were on at the time of this experiment. For our phase 1 plants, they were exposed to 12 hours of heat stress with collections every 3 hours. And phase 2 was exposed to 9 hours of heat stress with collections at 0, 6, and 9 hours. To look at what was going on internally in phase 1 plants without doing a sequencing project on them, we chose to do a catalase assay. And catalase is very, very important because it's a reduction protein for a particular reactive oxygen species that is catalase. And catalase is very important because it corresponds to peroxides. And peroxides are generated through lipid oxidation. And lipids compose the cell walls and membranes of the plant. It's one of the more predominant proteins in those compounds. And that's why it's very important. And there is a lot of literature evidence that supports that when plants specifically are undergoing heat stress, catalase is the most produced reactive oxygen species. A previous study in blueberries on heat stress that looked at the transcripts of reactive oxygen species over time, found that catalase transcripts were produced the most during that experiment. So it's a good indicator of whether a plant is under particular abiotic stress and an indicator of heat stress. We did this by extracting leaf protein from our phase 1 plant samples. We introduced hydrogen peroxide into the mixture, and then the catalase that was in the sample was able to reduce that peroxide to water. And we introduced a dye that interacted with the remaining peroxide, and we were able to use those numbers to calculate the concentration of catalase in the sample. We also wanted to look at the stomata of the leaves, which are like mouths that are on the leaves, and the internal structures of the stomata, the organelles. And in order to do this, we used our phase 1 plants to develop a technique for randomization and imaging and Z-stacking, which is how you get like a 3D picture of an internal structure. And we ended up using 5 leaves per time period and 10 images per leaf, and then 10 Z-stacks per leaf as well, per image. So in our phase 2, we put all of that selection to practice. And here is part of what we did. So this here is a mortar, and at the bottom is a grid. So our phase 2 plant leaves were very small, and we were able to dump in our sample for some liquid nitrogen over them to keep them from defrosting to prevent degradation. They would swirl around a bit, and we would use a random number generator to select a quadrant. We would then number the leaves within that quadrant, and then use a random number generator again to select the leaf. And we kept doing that until we had all the leaves that we needed for that sample, and then we repeat until we were done. On the right are these programs that we used for our imaging. This at the top is a program called Zen 2009. It was used for capturing our images as well as obtaining our Z-stacks. At the bottom is a program called Huygens. It was used to do deconvolution, which is light packing in order to clean up our images. And Emira's in the middle here was used to calculate the volume and surface area of those structures. And this is the microscope that we used to do this analysis. This is a Zeiss LSM 510. And all of this equipment and training for this part of the project was done at Delaware State University in the Oscar building in the imaging lab that's run by Mr. Michael Moore. And this is our transcriptome sequencing at phase 2. So we had leaf samples from our phase 2 plants, which is the corn blossom and Darwai, and we shipped those off to Virginia State University, and they thankfully reached the lab without degrading or anything. They were well packed in dry ice and all types of stuff. And we used the de novo sequencing in order to de novo assembly in order to assemble our transcripts, and we received back 2 annotation files, one for each species, and also differentially expressed genes for the time periods of each species. We were able to use that information that we got from the lab to do alignments to related species. And with that, we did a pathway analysis. From this pool of transcripts, we were also able to make primers to do real-time PCR. And this is how we did the pathway analysis. We actually did 2. We did a metabolic and biosynthesis analysis of the pathways. So in the annotation file that was constructed with the last 2 XGO, there's a lot of information in there, one of which is called an enzyme commission code or EC code. That code corresponds to a file in the Kaido Encyclopedia of Genes and Genomics, or the KEGI database. We were able to match the differentially expressed gene transcript information with the annotation file, and then get those codes. We put them into the KEGG database, and it gave us back a lot of information, including the metabolic and biosynthesis pathways associated with each gene. And from that information, we were able to look into what the plant was prioritizing and doing during that time based on what pathways were most up and down regulated. For pathway enrichment, we used 2 programs. Both of them are R packages in order to do them. We use Cluster Profiler, which relies on k-means clustering, and it's a technique that uses the false discovery rate and the p-value to make clusters of data that is this is similar, and it keeps doing that until those clusters do not move anymore. And then it gives us back a lot of information about those clusters, including what pathway IDs correspond to each one, and all the gene IDs that are associated with that pathway. We can then use that that pathway ID and plug it into a program called PathView, which uses information from the K database, some maps that it makes, and it lays our data on top of it so we can see what is up and down regulated in those enriched pathways at the time. In order to do this, we had to align our transcriptome data to 2 genomes. We aligned it to Vitis vinifera, which is the wine grape genome, and Arabidopsis, which is a heavily researched plant genomics genome. The reason why we had to do this was because currently there is not publicly available a genome for blueberries that is also associated with the NCBI database. And we needed those NCBI gene IDs in order to conduct this analysis, because that's what the programs relied upon. And so we use the wine grape genome because it's a very close relative of the blueberry, and also Arabidopsis for extra coverage because it's such a heavily researched genome. Finally, we did real-time PCR, and we selected 29 genes for to be made into primers. We selected 20 genes that were involved in highly expressed metabolic pathways. 10 were from corn bosom, and 10 were from DARY. We selected 3 genes that were upregulated in DARY but downregulated in corn bosom, and 2 genes that were upregulated in corn bosom and downregulated in DARY. And we also chose 4 housekeeping genes. These were all selected randomly from spreadsheets that we had of these genes. And then we used DNA. Once we got our primers back, we used DNA from phase 1 plants, which were blueberries that had a mixture of corn bosom and DARY and one diversifying plant. We used that DNA cocktail to check amplification. We then used cDNA that was made from RNA of corn bosom and DARY in order to check amplification again. Once that amplification checking was done, out of the 29 primers that we had, 27 amplified well. That's still a lot of primers for real-time PCR. So we then further selected for primers that amplified well over all of the time periods, and then used randomization to select 5 primers that were finally used for real-time PCR. And these are our results and discussion. So these are our plants that we used in phase 1. These plants are between 2 and 3 years old, and 5 of them have a mixture of corn bosom and DARY in them, except for 1, Prince Rabidii, which is a different wild-type plant. These are how they looked before they were exposed to heat stress, and this is after. It is evident that there is some discoloration of the leaves. Some of them look very similar, and this could be due to they're not direct blends of corn bosom and DARY. They're also mixed with other things, but it's somewhere in their background. But we also wanted to get an idea of what was going on internally. This is a normalized graph of the pathways concentrations in each of our phase 1 plants. And we saw 2 predominant trends. We would either see a large spike somewhere in the time periods, or we would see a gradual progression. And if there was a large spike that was towards the start of our heat stress, one thing that could be happening is that the plant senses that it's under stress, it turns on everything, and then it makes internal adjustments in order to accommodate the new conditions. Once those internal adjustments are made, it no longer needs that high concentration of metabolites, and it metabolizes them. If it's somewhere in the middle, it could be that it prioritized perhaps making adjustments first and then decided to deal with the concentration of peroxide by deploying catalase, or it could be that the concentration of peroxides had to reach a certain threshold before the plant deployed defenses in order to deal with it. For the gradual progressions, it could be that the plant just released catalase as needed based on the peroxide concentration that was present at the time. And these are our phase 2 plants. On your left is DARY here, and on your right is corn bosom. And these are the plants that were used in our experiment. And one thing that we were interested in are the leaves of these plants. So on your left is corn bosom, and on your right is DARY. And right off the bat, we can see that Corymbosum has these very nice broad leaves here, and Darwai has these smaller leaves. Darwai is a shrub, but it also has a little bit of a sheen to its leaves, like a waxy coat on top of them. And some of the leaves are actually also slanted. Some of them are very perpendicular as well. And those are traits that are associated with plants that have a heat stress avoidant mechanism within them. And that was very interesting to us. And we wanted to know what was going on the leaves during the time of heat stress. And this is what leaves look like under a microscope. These mouth-like things are stomata, and these are little orange lights are the internal organelles that we measure the surface area of. This one's kind of open, this one is semi-open, and this one down here is closed all the way. And so when we were doing our imaging, we wanted to measure the surface area of these red lights and also wanted to look at how close these mouth gaps here came to touching. And this is what one of our Z-stacks looks like when it's completely rendered. This is a program in Amiris. This is actually from my data set. And what it does is it uses the light information that we give it, and it reconstructs those structures, and it's able to take the surface area and volume data and give it back to us, and we're able to do analysis with it. We can also only select around certain regions, so we would like to select in this area. And this is our stomatic gap closure data information. This is how much the gaps came close together to touching over time. And the reason why this is important is because when a plant is under stress, particularly heat stress, water is being lost at an alarming rate. And so one of the instinctual things to do is prevent water loss. And one way to do that is to close stomata. But the problem is if the plant closes its stomata too much, it won't be able to photosynthesize because it can't take in carbon dioxide and deploy oxygen. And that is problematic for the plant. But we wanted to see what adjustments were being made during this time to accommodate the plant during stress. And so we can see blue is Corymbosum and orange is Darwai. And Corymbosum makes a very sharp closure, and then like close to 9 hours, it starts to open up again, where Darwai is a sharp, not sharp, but it's steadily closing its stomata. So what does this tell us? It tells us that the plant is making internal adjustments in order to accommodate the new conditions, and it has to make some choices in water retention and photosynthesis at this time. We can also see here, this is the surface area change of the internal stomata organelles. And under heat stress, especially, the cell walls and membranes are under attack. The electrons are very excited, and that can lead to denaturing. And if you think of it like a coin purse where all the coins are stacked in neat little stacks, and during heat stress, those stacks start to move and then they slide over. And if it slides out too much, the sack can break. And that's what's happening, and that is what usually happens under these excited conditions if they're prolonged for too long. So we wanted to see what was happening here. And the blue is Corymbosum and the orange is Darwai. And we can see that Corymbosum is holding steady while Darwai is expanding a bit. Now, intuitively, we would think that maybe Darwai is under more stress than Corymbosum. But another way to look at it is to say that during heat stress, internal adjustments need to be made in order to survive. And perhaps part of the internal adjustments that Darwai had to make was expanding its internal organelles to accommodate the excited electrons in order to continue to survive in these conditions. But this was not enough. We wanted to look at our transcriptome data. In order to do that, we had to have some really good RNA, and we did. We had written values in the low 6s and high 8s. Which was backed up by our very nice gel pictures, like clean and crisp RNA that was used to do transcriptome assembly, and we got over 200,000 assembled transcripts. One thing that we see here is that Darawai on this side and Quorum-Boson on this side. Darawai has more assembled transcripts than Quorum-Boson, yet Darawai has less differentially expressed genes than Quorum-Boson at the time. And Quorum-Boson just generally, it seems that during stress, its response has just increased. Like there are a lot of upregulated genes at 6 hours, and there's even more at 9 hours. And then in Darawai, at 6 hours, there is upregulation in the genes, but there's a very sharp downturn at 9 hours with a whole lot of downregulation. And to better visualize this is a volcano plot that shows a graph of the up and down regulated genes at the time. The red is up regulation. They think of it as the genes being turned all the way on, and down regulation is like all the way off, right? And so here, this on the top is Quorum-Boson at 6 hours, at the bottom is Quorum-Boson at 9 hours. And we can see kind of like this exponential type of response, not exponential, but we can see that it's increasing in strength here, where Quorum-Boson at 6 hours is still uptrending in its differentially expressed genes, and then at 9 hours, it's still doing the same thing. Whereas Darawai during the same time period, this is at 6 hours, where there is a huge upregulation response at 6 hours, but at 9 hours, we're seeing a lot of downregulation over here. So this really interests us because these responses on the surface look very different, and we wanted to know what was going on internally with these plants. This is some of the results from our metabolic activity pathway analysis, and it's very hard to see here, but the most up and down regulated pathway was the metabolic pathway, which needs to be in equilibrium in order for the plant to survive, because it controls a lot of things, one of which is the cell wall cohesion, which is very vital in order for continued survival. One thing to note that was very interesting is that despite Quorum-Boson have many more differentially expressed genes than Darawai had a higher diversity in metabolic and biosynthesis pathways. And it's easier to see here, where this is the biosynthesis pathways in 6 and 9 hours of Quorum-Boson, and this is the biosynthesis pathways in 6 and 9 hours of Darawai, where Darawai has a much higher diversity in the pathways that it engages in Quorum-Boson. So this could tell us that perhaps Darawai is performing the same task with less genes and engaging more pathways, which is quite interesting. So we wanted to see what the enrichment pathways look like. And this is our general results from that analysis. And one thing to note is that Quorum-Boson at 6 hours had the most active pathways during that time period, and there was no other time period or any other species that even came that had this amount of active pathways. For the big gene ratio, what this shows is the amount of genes in our data that correspond to a particular pathway, or out of all of the genes in the universe of that genome that could possibly correspond to any pathway. And our p-values were very significant. One pathway that showed up in every time period and in both species was the protein processing in the endoplasmic reticulum. And that is a common pathway to show up in heat stress reactions, but what's interesting is what was engaged in those pathways. And this is Quorum-Boson at 6 and 9 hours in the protein processing in the endoplasmic reticulum. The red is up regulation and the green is down regulation. And what we can see here is that heat shock proteins are being engaged. That is a normal heat stress response thing. Quorum-Boson at 6 hours is capturing misfolded proteins, and it's engaging a few genes that are reinforcing the cell walls and membranes here. At 9 hours, we see a lot of down regulation, but the genes that are being down regulated are responsible for capturing misfolded or malfunctioning proteins, which those, if they are allowed to prolong and go through the plant, they can cause a lot of issues. But we do see more upregulation in proteins that correspond to reinforcing the cell walls and membranes at 9 hours. At 6 hours, it's very hard to see here, but all of these pathways are doing the same thing. They are breaking down materials in order to create items to reinforce the cell walls and membranes. Corambosum is engaging 3 pathways with that sole function to just reinforce the cell walls and membranes. That's quite significant because at 9 hours, part of those pathways are still engaged. Here, these genes are still making materials to reinforce the cell walls and membranes. And it's still capturing proteins and amino acids as well in order to do it. One thing that is very significant here is what's down-regulated, these 2 green boxes. These are proteins that correspond to reactive oxygen species reduction proteins. They perform a similar function to catalase. And the reason why that's significant is because that means that there are harmful chemicals at this time that are building up in this plant. It's evident that they're there because those 2 particular boxes are down-regulated. And that means that that corambosum is intentionally allowing these harmful chemicals to build up in its system and prevent itself from dealing with that in exchange for reinforcing its cell walls during this time. So what we can see overall is that at 6 hours, corambosum is very focused on capturing misfolded proteins and also reinforcing the membranes of its cell walls and reinforcing membranes and cell walls. At 9 hours, we see that it's still continuing to do that, and it's breaking down a lot of materials in order to do that, and it's even down-regulating its response to reactive oxygen species in order to facilitate that response. For DAR-Y at 6 and 9 hours, we see something very different. We still see the heat stress response, and we also see the capturing of misfolded proteins. But what's different here is that DAR-Y is engaging proteins who's responsible for capturing mutated mRNAs. And it's also engaging proteins that changes the chemistry of the cell walls and membranes. Not reinforce them, but changing the chemistry of them. And that is very different. But what is very interesting is that at 9 hours, it seems to have succeeded because it's down-regulated everything. It's turned off the entire heat stress response, except for this one lone upregulated gene here, which is responsible for capturing misfolded proteins. At 6 hours in DAR-Y in the ribosome, we're seeing something very similar to the tune of the mRNA capture, where it's capturing our mutated and damaged instructions for protein generation. So if it captures the mRNA, which corresponds to how to fold a protein, if that mRNA is damaged, then the likelihood of it producing a damaged protein is increased. But if they're capturing that beforehand, then that likelihood somewhat, that just provides some quality control. And here in the ribosome, we're seeing that same thing where a lot of mRNAs are being captured, ones that are mutated here. But also, we see that parts of this ribosome correspond to particular types of proteins, like proteins to make a vacuole or to make a certain type of item for a cell. And it's intentionally trapping those mRNAs, those instructions, in order to allow different types of mRNAs, perhaps ones that it needs more than others, to go through and be folded in the protein. At 9 hours here, we see that DAR-Y has engaged its photosynthesis regulation genes. And that is very, very exciting and very significant because plants that are heat stress tolerant are able to still photosynthesize over during the induced stress period. And up here, this is photosystem 2, this line here, and this bottom line is this top line is photosystem 1, and this bottom line here is photosystem 2. As we can see, photosystem 2 is highly enriched, and parts of photosystem 1 are. And these proteins are performing a similar thing. What they're doing is they're monitoring the electron activity in each photosystem. And if one photosystem appears to almost be overwhelmed with the amount of the electrons it has, it will kick that electron over to the other photosystem. So it's managing the amount of electron activity between them so that neither get overwhelmed and they're able to still continue to photosynthesize during this time period. One thing that we saw is that all of these alignments were done with the wine grape Arabidopsis for confirmation. And we saw an extra pathway in our Arabidopsis alignment that we didn't see in the wine grape genome, but it does confirm what we're seeing here. And that is this pathway. This is the circadian rhythm pathway in plants. And what this pathway is responsible for is regulating photosensitive genes, ones that come out at certain times in the day when there's certain light requirements are met. And it's very interesting because this is a blue light protein that is responsible for being produced for capturing lots of blue light if there's too much present. That's interesting because here in photosystem 2, all of these proteins correspond to blue light. And so this confirms our findings for our previous map. But what it also does is tell us that up here, this protein is produced for UV protection. So not only is Darwin able to continue to photosynthesize and also manage the electron activity, it's also able to produce proteins that protect itself from harmful UV rays as well. And so we see that in Darwin at 6 hours, it's more focused on filtering out damaged and mutated mRNAs and also altering the chemistry of the membranes and cell walls. At 9 hours, we're seeing that there was a great downregulation in those heat stress proteins, which tells us that the adjustments were successful, and Darwin was able to continue photosynthesizing and also regulating the amount of electron activity. And this is the results from our real-time PCR. The blue is the transcriptome data, and the orange is from our real-time PCR. And largely, our real-time PCR trends in agreement with the results from our sequencing. And so, in conclusion, we see that chlorambucil is reinforcing its lipid membranes, which is the cell walls and membranes, and Darwin altered the chemistry. And chlorambucil was still repurposing items in order to do that, while Darwin was able to move on to photosynthesizing and other things. And so one thing that would be nice to look at going forward is what mechanism is responsible for the change in lipid chemistry over the reinforcement, and how to screen and identify if that a mechanism will be engaged in order to tell which plants will do better in those heat stress circumstances than others, doing some type of markers for that. So I'd like to take this time to thank many people. I'd like to thank the people in my lab, the molecular breeding lab, especially Dr. Leva for being my advisor. I really appreciate it very much. And I'd like to thank Aisha, who as an undergraduate student here woke up very early at 5 a.m. to help me log plants into the lab for adjustment periods. And also, even as a graduate student now, she's still helping me with my research, so I very much appreciate it. I'd like to thank the people in Dr. Cow's lab, the plant genomics lab, for allowing us to use their equipment and sharing their expertise. And also, especially thanks to Dr. Todd, who was very nice to me when I came to her with a bunch of pathway maps, and she taught me how to read them, and also helped me organize my thoughts and do my analysis. I very much appreciate it. At Rutgers University, our collaborator, Dr. Borsa, for not only providing the plant material for this project, but also for teaching me how to propagate blueberries. And that was a skill I was able to share with my lab mates, so I thank him for that. Thank you to the Graduate Student Association here in the Agriculture and Natural Resources Department for being very encouraging and providing a lot of resources for new students. Thankful to my family and friends who are here not only to help me, but also are very encouraging and very understanding of my schedule during this time. Thank you guys so much. Thank you to my committee members who I could not be defending up here without their service. Thank you so very much. I very much appreciate it. And thank you to the Delaware State faculty, Dr. Marsh, who's the dean of our department, is very encouraging. Dr. Smith, who advised in the randomization of the leaf experiment, thank you so much. Mr. Michael Moore, who provided not only the imaging facility, but also the training, and he also made that clip that you saw there of the stomata moving around. And thank you to Dr. Scott and Dr. Smolinski, who advised in the bioinformatics portion of this project. Thank you all very much. Thank you, and are there any questions?
So if somebody wanted to take your research to the next step, continue on with it, what direction would you suggest that they go?
So I would say to look more at the mechanism that's responsible for altering the chemistry of the walls and the membranes versus the reinforcement, because Coram boson was still trying to reinforce and it wasn't able to get past that in the time period of our experiment versus Darawai, which was able to change the chemistry and continue to photosynthesize, and that's very important for heat stress tolerant plants, because the time that Coram boson is taking to reinforce its membranes, it's also allowing itself to be damaged quite a bit. And so looking at what is responsible for making that decision would be very helpful. So that's what I would do next.
I just had a question about the berries, because you have the Darawai says a bush, it's a small bush, right? It's from Florida.
Yes, it's a low bush.
And then is the other one, it's taller, right?
Yeah, it's a high bush.
So is there a difference between the berries themselves? Is one small and one large?
Yes. Good. Okay. Darawai tends to have smaller berries and they're very tart. They have a high vitamin C content. Coram boson has really nice big berries that tend to be juicier and a lot sweeter. And so consumers tend to like the juicier berries and really like the sourness. So that's part of the reason why the Darawai berries aren't as popular in commercial markets. And so we still need to maintain market appeal and also make the plant stronger. So that's part of the reason why this is very important.
And the root structure. So what's the difference? There must be a relationship between the root structure of each of these plants and the leaves, because you're growing them in pots, right?
Yeah.
So how's the root structure related to the leaves? Is it different or?
Unfortunately, that is outside of my.
The Darawai is a rhizotomous roots. Coram boson has a more of package system. And Jodi should have mentioned that the commercial blueberries that we get from the shops, most of them are tetraploids. So we have polyploid, they are polyploids. The research that we are doing now with this Darawai and Coram boson, they are diploids.
Yeah.
So having diploid status helps us for genetic analysis much easier.
Thanks.
Jodi, I like your statement that you said we've been breeding plants for a long time, and then they probably have lost some ability to respond to the environment, right? Or something like that. So you may know that, for example, in tomatoes, because of our breeding for the large fruit and all of that, we've lost the taste gene in there because they were intricately linked, okay? It's a Cornell University study, you should look it up. So, is there something in blueberries that you think has been lost by breeding and that needs to be brought back?
One trait that seemed to be lost in blueberries was their diversity in the environments they could grow in. Blueberries used to be able to grow in forests and marshes and other areas like that. But now blueberries have been so domesticated that they can really only grow in very specific pH soils. So they had a higher diversity of the types of pH soils that they could grow in. They were still very acidic soils. But now it seems that from the earlier studies till now, there seems to be an increase in the acidity needed for the blueberries. So bringing back some of that diversity would help with that diversification from my readings.
Keep up with your plant breeding goals down the road in life.
And
Okay, well, first of all, I think it was a wonderful presentation. But I'm going to ask you two questions which might be outside of the scope of your work, but it has extension implications. First thing, how does heat stress influence the quality and quantity of the fruits that those plants get generate?
So heat stress, prolonged stress can stunt the plant to where it will have reduced yield or no yield in the blueberry plant. So right now, there are places in the Midwest that aren't seeing any yield in their blueberries because it was hot very early on in the season and the plants never quite recovered and they weren't able to produce.
Right. So the thing is, even if you get blueberry fruits, does it influence the quality, the taste, texture, storage ability and stuff, of the fruit? Does the heat stress actually influence the actual quality of the fruit itself?
For blueberries specifically, that's a bit outside of the scope of my project, and I don't know the answer to that right now. But I can look it up and share that with you. Or you can check back with my lab in a few years and they'll have a study on it.
So, last question. So, the thing is, what kind of so the actual research that you did, how do you think that farmers can really benefit from the research aspect of your what kind of information can farmers get down the road based on the research that you get for farmers who experience problems because of climate change?
Right. So right now, farmers can look at some of the physical signs of heat stress avoidance. So like the smaller leaves, the waxy coat, the orientation of the leaves, in order to select plants that are more tolerant, right? Like today, that's what they could do today. Down the road, what my research will enable others to do is to develop markers and indicators for stress tolerance, and then they'll be able to breed plants specifically that have that trait down the road.
Thank you.
they are satisfied with 1 million.
Huh?
So
Satisfied,
just to add,
but you can add it,
just for additional information, the blueberry breeding programs mostly relies on not only the excellent funding from the competitive grants, from the growers as well. So far, where we have strong breeding programs, those states, except California and Florida, those states also have low soil pH condition. So the growers don't want to fund breeding programs that they want to develop varieties that can withstand high soil pH. That's one condition. So that's why I'm not getting funding from growers.
Right.
And for our state, this is a good trait to explore. And then same with for high heat stress tolerance. So probably down the road in 2 years, we may have, like Jodi mentioned, few traits or few varieties that we have not explored yet. Those varieties can be a good one for farmers to understand.
Thank you.
Any more questions?